So thank you to David for the introduction, and welcome everyone to this first talk in our first session entitled Variation in the Human Genome. And this is an outline of what I'm hoping to cover in the next 20 minutes or so. So we're going to learn about the extent of variation, as well as different sources and mechanisms of variation. And then in my second short talk, I'm going to cover what variants are important, how do we find out which variants are important, and what resources are available to help us with these questions. And both these talks really build on the pre-course talks that were given by Andrew Reed and Hans Hennies. So if you haven't had a chance to look at those just yet, I would really encourage you to do so. So the extent of variation in the human genome is huge. The human genome is made up of 3 billion base pairs, including up to 25,000 genes. And each gene codes for, on average, 3 proteins. And so far, more than 80 million sequence variants have been identified. So given this huge extent of variation, it's very important that we have this remarkable accuracy of duplication. And this is largely because of base pairing, the way that DNA replicates itself, but we also have these mechanisms of proofreading and mismatch repair. So DNA polymerase is the enzyme that can correct single base pair mistakes. And then there are separate mechanisms that allow longer sequences to be repaired when mistakes have occurred. So another thing it's important to remember is that only about 1 or 1.5% of the whole genome is protein coding. So if we think of the whole genome as this apple, the exome, or the region that actually codes for proteins, is only a small slice, about 1% of it. And when it comes to understanding variation, it's kind of humbling to remember that 60% of genes are conserved between the fruit fly and the human. And we know that about 99% of the human genome is the same as a chimpanzee genome. And more recent studies have shown that we're probably even more similar to gorillas than we are chimpanzees. And let's just pause and think about different sources of variation. What causes so much variation between different genomes and different people? So focusing on the DNA, we've already said that the exome is the protein coding sequence, and changes in protein coding can obviously produce changes in proteins. But the non-protein coding sequence is also a very important source of variation. And some people have previously described this as junk DNA, but we know that 80% of the human genome is transcribed or binds to regulatory proteins or is associated with some other biochemical activity. So it's clearly not just junk. And this has been really illuminated by data from ENCODE. And these non-protein coding sequences contribute non-coding RNAs, of which there are many different types. We've got untranslated regions of messenger RNA. We've got introns, which are regions within the gene. We've got pseudogenes, which might have a similar sequence to a functional gene, but appear to be either non-functional or have a different function. And then there are regions of very repetitive DNA sequences for which the function is unclear. And then there's regions of regulatory DNA, and a large amount of work has gone into understanding these regulatory regions in recent years. So other evidence of important sources of variation comes from identical twins. So in theory, these beautiful identical twins have identical DNA, but clearly they don't have an identical phenotype even at birth, let alone later in life. And these additional sources of variation might come from copy number variants that are harder to detect. They might come from mutations that occur in utero, and they might come from epigenetic effects that can accrue over life, as well as obviously other environmental effects on the twin phenotypes. So epigenetic modifications to DNA take the form of methylation or histone modification. And these are really dynamic processes. So epigenetic modifications can be affected by multiple different environmental effects, and they can occur anything from in utero to really throughout the lifespan after birth. So coming back to sources of variation within the exome or the protein coding part of the genome. So here, single nucleotide changes are the most numerous, and we call these single nucleotide polymorphisms or single nucleotide variants. Those terms can be used interchangeably. In addition to single nucleotide changes, we can have larger insertions or deletions where DNA is added or removed. We can have translocations where a chunk of DNA can be moved to a different part of the genome and therefore have different effects. We can have inversions where the DNA code is kind of flipped over or reversed. And then we can have copy number variation. And what this describes is where a region of DNA can be duplicated and different genomes can have different numbers of these duplications. So taken together, these are called structural variants. And this structural variation is harder to detect sometimes from straightforward sequencing. Sometimes more specific analysis is needed to make sure that you don't miss the structural variants. So I've explained already that exome sequencing, whole exome sequencing, aims to sequence this portion of protein coding DNA. But it's important to remember that whole exome sequencing could be better described as whole exome sequencing without the W, because there are always holes in the data. There's always missing data, even though we're trying to capture the whole exome, we don't succeed in doing that. And it's important to be aware of this, because if you don't find a variant that seems to cause the phenotype, it could be that just you've missed that bit of sequencing information. So looking now at the whole genome, here the whole genome gives information about control of gene expression. So not just the genes themselves, but how those genes are expressed or controlled. And there are multiple complex mechanisms, and you'll learn about several examples of these throughout the course. And it's important to remember that the control of gene expression is tissue specific. So we're lucky that we can study skin, it's more visible, we can also sample it. So we have more opportunities than many specialties to get a grasp of the tissue specific genetic effects. And there is also a whole world of RNAs, so not just messenger RNAs and transfer RNAs, there are long non-coding RNAs, there are microRNAs, and multiple different small RNAs. And again, you'll learn a lot more about these as we go through the course. Cancer genetics has offered a unique insight into more severe genetic variation, including large deletions, tandem duplications, and then these catastrophic events that have been demonstrated in cancer cells called chromathripsis, when whole chromosomes can be broken apart and then knitted back together in a different order. So listing the mechanisms of variation now, as opposed to moving on from the sources of variation to potential mechanisms of variation. So we've spoken about single nucleotide and structural variants. We can distinguish somatic variation, which is a variation occurring in the tissue, as opposed to germline variation occurring in the inherited DNA. We need to be aware of mosaicism and revertant mosaicism, where we observe different genetic effects in different cells within the tissue. And again, you'll learn more about this in the talks. DNA damage is an important source of variation that accrues through life. Not to be forgotten, especially in skin that is at the interface with a lot of UV damage and other environmental effects. I've mentioned about gene and protein expression in different tissues. So control of gene and protein expression contributes to variation, and this can be context specific. And there are tissue specific factors. So again, in skin, we're very fortunate to be able to study tissue specific events in our tissue of interest. And then splice variants, which you'll have heard about in the introductory talks, add an extra layer of complexity. And again, to re-emphasize, these are all dynamic processes. This isn't something that's set in stone at one point in development. These can occur throughout the lifespan. So in general, common variants are thought to cause common disease, and rare variants might cause more severe, rarer disease. And common variants were previously thought to have small effect sizes, rare variants thought to have large effect sizes. So these assumptions are based on the frequency of DNA variation in the population compared to the penetrance of disease. So rare variants often are highly penetrant, causing classical Mendelian disease, whereas common variants were thought to be lowly penetrant, often identified by GWAS as contributing to common complex traits. But the more we've learned about different variants on a population scale, the more it's become clear that actually this is rather a false distinction. And we know that even common variants can be highly penetrant, and common variants can come together to cause severe disease as well as milder disease. So, aside from the fact it's very interesting to understand how DNA works, and obviously understanding variants is intrinsically important, we can use an understanding of genetic variants as a tool to understand mechanisms. And the best example of this is Mendelian randomization, where genetic variation can be used to assign people, similar to a case control study, assign people based on genetic variants that are present at birth to test whether those variants are having a causal effect on disease. And again, you'll learn about Mendelian randomization later in this conference. Another important use of understanding variants is that predicted loss of function variants are a natural in vivo model of human gene inactivation. So it's a really good way to understand how genotype leads to phenotype by studying humans that have those mutations. Gives us information about the gene function. And recently it's also been used to guide drug discovery. So if you look on a population level and find that variants, for example, variants in this LRRK2 gene in the population are common and are not associated with the disease, that gives us confidence that we can design drugs to block that gene, and this is predicted not to have a detrimental effect, so those drugs are hopefully going to be safe. And the example given here are LRRK2 blockers that are going to be used for treating Parkinson's disease. So, to summarise what we've discussed so far, there are multiple sources and multiple different mechanisms of variation within the human genome. Recent technological advances have really brought about a massive expansion of understanding. And this increased awareness of the extent and importance of variation in coding and non-coding regions is important for research and clinical application. So, how to make sense of all this variation? Now that's challenging, and that'll be the topic of my next talk. Thank you.